So this video is about counting cells and microbes. It's about the enumeration. Counting cells and microbes is really important in many different areas. Depends regardless of what you work on. Whether you work in the brewing industry trying to count yeast, whether you're in agriculture trying to count mycorrhiza, whether you are trying to develop a new vaccine, whether you're trying to develop new antibiotics, or any other way of that you're trying to examine how things change over time in terms of growth. That's all how we enumerate microbes. And there are many different ways, really cool ways, lots of modern ways. But I'll show you in this little video 3 different ways of how we explain to you how to count cells and microbes. Alright, so there are different types of methods of measuring the numbers of cells. First of all, there are direct cell counts, where you can count actually each individual cell. And then there are indirect cell counts, where you count the population of cells, for example, the turbidity, how dense it is, or end products, how much end product is used. So there are different ways of counting either individual cells or populations. It also, for the direct cell counts or for the cell counts, it also makes a difference whether you count live cells or whether you can count populations. For example, when you plate cells and look for CFU per mil, the organisms have to be alive to form a colony. But for example, if you're looking at optical density, it's just scattering of light. So the scattering of light doesn't know whether the organism is alive or not. So there's a lot of different factors to be considered when you are enumerating cells and bacterial populations. In this short overview, I want to talk to you about several different methods. The main thing is the plate count and serial dilution, which is sort of the pet peeve of microbiologists. It's used all the time, everywhere. The optical density measurement for growth curves, and also the hemocytometer counting for cells and yeast. So the different way of direct cell counts are hemocytometer, which is a glass slide and you put a suspension of your cells and you're actually physically counting individual cells and then multiplying it to understand how many CFU or cells per milliliter you have. And you can distinguish, depending if you're staining it or not, you can distinguish between live and dead cells. The plate count, the serial dilution, is where you're actually looking for live cells. So you're having a sample, whether it's ice cream or anything you want to look at, diluted usually tenfold, and then plated out in agar plates. And that only works with cells that are live. So you're looking at a plate count, it counts live cells in a specific sample, and that's one of the most common methods. Flow cytometer also determines each individual cells, and that's determined by light beams, where you stain your cell with specific dyes, fluorochromes, and that are detected by a machine, by a sensor. And then you can distinguish different populations depending on their stain. Very powerful method, very commonly used. Indirect cell counts are determining the concentration by more physical properties, like for example optical density or kinetic end products. So for example, optical density measurements is when you do a growth curve and we just look how cloudy, how turbid a population gets, a culture gets over time, and how long does it take to get to the turbidity and how steep is the slope. So we'll talk about that in a little bit. And then there's end product or kinetic reactions, which can also give you an indication of the numbers of cells present. So the main topics I'll talk about briefly is the hemocytometer counting, the plate count and serial dilution, and the optical density determination of the growth curve. The first explanation what I want to give to you is serial dilution in spread plates. So you take a sample, maybe it's contaminated orange juice, and you do 1 to 10 dilutions, and then you So serial dilution or spread plates is a method where you take an original sample, you dilute it out, and you plate each dilution on a different plate, usually in replicates, to see what are the CFU per minute, what's the concentration per milliliter in your sample. And let's talk about contaminated orange juice. So you say you have contaminated orange juice, and you take 1 milliliter of the orange juice and put it into 9 milliliters of your diluent. So let's make a 1 to 10 dilutions, 1 part and 9 parts, and repeat that several times. And then from each of your dilution tubes, you plate out 100 microliters on your agar plates. You let it grow up and then count the colonies. You always obviously want to have a control to make sure your diluent is clean and your experiment is going well. So what happens and how you can calculate it is you're counting that one plate that has between 30 and 300 colonies. You count the number of colonies and then you multiply it by the inverse of the dilution factor, considering how much volume you put on your agar plate. So for example, in this case, if you have 83 colonies on your 10 to the minus 5th dilution and you only plated 100 microliters per plate, you calculate 83 times 10 to the 5th times 10, because your concentration was per 100 microliters, and your total concentration of the contaminated orange juice would be 8.3 times 10 to the 7th CFU per ml. A similar experiment can be done with petrifilms. Petrifilms are the same thing as agar plates, except they're dehydrated, and you add 1 milliliter per plate on your sample. So again, the same experiment. If you add something contaminated, then dilute it out tenfold. And in this case, you have 10 to the minus 8th plate that has 33 colonies. So you just multiply 33 times 10 to the 8th, which would be 3.3 times 10 to the power of 9 as your final CFU per ml in your original sample. Make sure that your controls are clean, because if you have contamination in your control, the experiment didn't work and you have to repeat it. That's a pretty simple method, very commonly used. Right, let's look at the spectrophotometer, or the growth curve. Growth curve can be measured in two different ways. You either look indirectly at the optical density, where you just look at the cloudiness over time, which can be measured by a spectrophotometer, or you take a sample at different time points, dilute it out, and then measure the CFU per ml with a diluting plates, what I just explained before. Both of them can give you similar results for growth curve analysis. And in one case with the spectrophotometer, you get an optical density measurement, and if you plate it out, you get a CFU per ml count over time. And there's a great software simulation where you can actually pretend to do growth curves under different circumstances, different conditions, and with different microbes. So check out that resource, and you can actually get the data as well. And what we want to teach you is to use these numbers and calculate the growth curve, the growth rate. How long does it take for a population to double? So get the numbers and then apply this formula to calculate the growth rate of your organism. And that's how you do it. So you have the growth rate, you have a lag phase, you have the log phase where it's the steepest growth, and then you have the stationary phase. The growth rate is calculated always at the steepest, at the fastest phase of growth of an organism. On the y-axis, you either have optical density or CFU per ml. On the x-axis, you have time. And you want to see how long does it take for the population to double, like in this case, to go from 0.4 to 0.8. How long does that take? And so you can just really eyeball it and go over on your axis and see, so at my final destination at 0.8, it's about 3.5 hours. In my initial time, when I had half the numbers of cells, it was about 1.5. So what's the difference between T2 and T1? So it's about 2 hours to double the number of cells. And that's how you can eyeball it. You obviously have to use the formula and use Excel to calculate it, but it's a pretty simple way to sort of estimate how long does it take for your population to double. And again, it's really important for applications like drug development and many other applications, food safety, to know how long does it take for organisms to double. All right, the third method of what I want to introduce you today and how to count microbes is the hemocytometer. And the hemocytometer goes back to the beginning of hematology, where physicians try to look at blood cells, red blood cells and other blood cells, to see if there's any diseases that can be detected by looking at the blood. And it has been more sophisticated and improved, and today we mainly use the Neubauer hemocytometer, that is a glass slide, a thick glass slide that has etched different squares and patterns on it. So if you count a certain area, that gives you a certain volume of cells, and that allows you to calculate the numbers of cells per minute. This is commonly used when you do tissue culture, when you dilute cells, but it's also used for brewing, where you want to see how many yeast cells you have per ml, and so on and so forth. Again, the simulation here is very easy, where you can look at different scenarios, different dilutions, and then you get a picture where you can actually count the cells. The way you count it, or the way you load it and use it, is you have your hemocytometer, you have a cover slide on it, and then you add 10 microliters of your diluted suspension of cells. And then you look under the microscope, and you can actually see this grid where you have different areas and different lines to count cells. Each area is a specific volume. So if you know the volume of the area and the numbers of cells, you can actually count the CFU per ml, or how many cells per milliliters you have. So for example, if you dilute your sample 1,000-fold, and if you have one square where you have 15 cells and you average the different squares, you just multiply the 15 cells by a 10 to the 4th of the volume of the square, and then you multiply it by the dilution you had of your original sample. Right, we'll explain that more in detail in another module. Right, so this really wraps up the different ways of counting microbes, what we teach you, which is the dilution plate method, the growth curve, and the hemocytometer. Fairly simple, but again, very, very powerful methods for you to know to work in any microbiology lab, in the clinics, in the diagnostic labs, or anywhere else. I hope you get the main points, and it's not really hard, so I'm sure you can get it.